I mean, it is so remarkable if you try to look at how the protein synthesis occur, and that's why the Nobel Prize, I think 2009 Nobel Prize, if I'm correct. So
Was it this recent that we kind of cracked down the protein synthesis process?
Yes. The genetic code was kind of cracked down, that how many amino acids are going to are there, and how the DNA is being, or mRNA is read into what amino acid. But the overall structure of ribosome was discussed now, and this is from one Indian or Asian scientist.
Welcome to the first episode of the podcast. Today's guest is Dr. Monika Sharma. Dr. Sharma holds a PhD in Computational Natural Sciences from IIIT Hyderabad. She's currently an assistant professor at Plaksha University, having earlier worked at institutions like IISER Mohali, Max Planck Institute of Biophysical Chemistry in Germany, Michigan State University, and the National Institute of Neurological Disorders and Stroke in the US. Her research focuses on the application of computational methods to understand biological processes, with a keen interest in the integration of deep learning with the exploration of conformational dynamics. Her vision for the future is to be able to create a world-class premier research environment where she can conduct biomedical research that will impact society as well as the healthcare industry. Let's go into the conversation. Let's start by defining what is computational biophysics and what and how of the computing, biology, and physics are coming together.
Okay, so as the name itself is computational biophysics, so it's like we are applying the physical principles by making the computational models and to understand the biological processes that sum up what computational biophysics is.
Okay. Yeah. So can we talk about like some of the main principles we're borrowing from all of these disciplines that are key to the confluence of these coming together in computational biophysics?
So there are various principles, and depending upon the problems and the applications you are applying to answer that problem. So there are various processes, or let's say I'm going to say, for example, all these biomolecules are chemical in nature, right? So they are going to be interact with each other. And those interactions you can kind of formulate in way of whether they have charged interactions or not, or whether they have some van der Waals interactions or not, which we call as hydrophobic and hydrophilic interactions. And the thing is that not only within themselves, but within the environment around themselves as well. So you can actually formulate those processes. So I can, let's say, use the Coulombing potential to calculate how the charges look like, how they're going to come together or repel each other, combined with the other forces, for example, van der Waals forces, that how the hydrophobic interactions they are going to come together, and that will be edited to give you some energy form.
Right.
So that energy form is going to tell you how the molecule is going to behave in the next step. So this we call as molecular simulations as well. So that's a one small instance where it is being applied.
And so the physics part of it is going to be about like the structure formulation of the molecules and
formulation of the processes, the interactions, the model of this. Okay, and the computation part will come, for example, if I have created over, let's say, the simulated, how I'm going to simulation over a time. I start from, let's say, time equals to 0, how I'm going to integrate over it. This is where the computations come.
Got it.
So this is with respect to the biophysics itself. The other is, for example, the AI, deep learning method and everything. So those can be applied to the biology as well. So coming from algorithms can come from any stream. But
got it, yeah.
It can be applied
we'll get into some of those. Can you also just throw some broad light on the subfields?
So the subfield are biomolecular simulations, drug designing, and then this is AI applications, and then the next will be computational genomics, for example, transcriptomics and metabolomics. So you can do computational as well. So these are the subfields which
so cool.
The broad subfield.
I think we'll get the chance to like delve into some of these through our conversation.
Okay.
Let's start from I think one of the most fundamental ideas here would be to just unpack protein synthesis. I think that's also like the central dogma. So can you just like briefly speak about the process of protein synthesis, and through this we'll also be able to establish the whole sort of hierarchy of cell, nucleus, DNA, gene, RNA, and splicing and all of that, all the way up to like protein folding.
So central dogma in biology.
Yeah.
So central dogma in biology is something which relates that you have a DNA molecule.
Yeah.
DNA molecule is going to be read into an RNA molecule, a messenger RNA, and that is going to be then converted into protein.
Right.
So that's the process which happens, and this is conserved across all the species. That's how you get. It's really hard to get. You won't be getting DNA to a protein. You'll be having an intermediate RNA and then convert it to protein.
Right.
So that's the central dogma.
And can we speak about protein synthesis? Can you talk about that process?
So protein synthesis is when you have this mRNA protein. So what happens is that in eukaryotic cells, if we say, I mean prokaryotics are easy. So these are something like we can think about the first cell which came up. You take up everything, and now you have like bacteria, E. coli. In those cases, what you have is that you have DNA which is lying in the cytoplasmic cell, which is not within a nucleus. We are the eukaryotic one, the advanced level of prokaryotic, I'll say, in a sense that here everything is compartmentalized. Okay, so you have a nucleus. That nucleus has a DNA.
Right.
Now the DNA is safeguarded that it cannot go out.
Okay.
In prokaryotic, the difference is that the DNA can be read and converted into RNA, and then it can be converted into protein. In eukaryotic, the DNA is not coming out, but the information need to be passed out. So what happens is that the mRNA is being generated. We call this process the transcription process.
Right.
So as the mRNA is being generated, then since it's a single-stranded mRNA and it can go out of the pore of the nucleus, so we call it nuclear pore. And once it goes out of the nucleus, so then the ribosomes, which are the protein-making machineries, so they can read the information which is kind of encoded
in the RNA.
In the RNA itself, because when you take a DNA, it is going to be transcribed, mRNA is going to come out, and then within the mRNA, it's a kind of a encoded information which needs to be decoded into protein. So then the decoding machinery, if we think, is a ribosome. It knows that what are the other partners I need to bring into to actually create proteins. So then the whole machinery came, and then the protein synthesis was done. I mean, it is so remarkable if you try to look at how the protein synthesis occur, and that's why the Nobel Prize, I think 2009 Nobel Prize, if I'm correct. So
was it this recent that we kind of cracked down the protein synthesis process?
Yes. The genetic code was kind of cracked down, that how many amino acids are going to are there, and how the DNA is being, or mRNA is read into what amino acid. But the overall structure of ribosome was discussed now, and this is from one Indian origin scientist. So Professor Venki, he was one of those who got the Nobel Prize for ribosome.
So in the protein synthesis process, at what points are we able to intervene?
Okay, so as a researcher, we can, let's say the one who is a biochemist, or let's say even a molecular biologist, so they can intervene in a sense. We can mutate proteins synthetically. So you take a protein out, you can encode in an E. coli, and then you create, or any other vector you want to convert, and then you can have a mutants of proteins done. So that can be tweaked. One part is that. The other part is that currently the CRISPR-Cas which has come up. So that can also help you tweak at the gene level to actually convert itself.
So that's at the gene level, or is it at the splicing level?
Okay, so CRISPR-Cas can be at the gene level.
So that would mean editing at the gene level, and so that would like then result into a different kind of RNA itself.
Guided mRNA, and then that can be converted into protein part.
And the other point of intervention would be that you get into splicing of a RNA.
So splicing is something, splicing is something you have in mRNA. mRNA is a very transient molecule. So working with mRNA is a bit difficult in a sense. It's a very, very unstable kind of protein, not very stable as DNA part. So the splicing usually occurs in a cell itself. Though we can change some changes in the genetic, we can make, let's say, if we knock out where the splicing is going to happen, one person can have another protein to be done. Studies are still going on in that sense. It can be studied, but it's the work in progress.
So you're saying that
Proof of concept can be
of
there.
the transient nature of the RNA, it is one of the main challenges is that it's difficult to work with.
Yes.
So there's a natural machinery which is involved in like splicing the RNA, but we necessarily don't really have a lot of sort of like control on how is the splicing happening.
We know a lot deal about it, that where it might be happening, how we can do that, but kind of modulating that, those kinds of we'll call sensitivity in the experiments is still missing.
Got it. And so I think like one of your areas of research is also alternate splicing.
Yes, so I'm actually looking at trying to look at one aspect is that we know that there are certain parameters in mRNA which can be looked as the tags or the kind of flags that here the alternative splicing is going to happen. And my research is that how the structures of those tags in partner with the proteins, because here also mRNA is being influenced by the protein itself.
How is mRNA already influenced by the protein?
Isn't
the mRNA going to result into a protein?
So there will be different mRNA to create that protein.
Okay, right. So there's already an existing mRNA.
There is already an existing
that is interacting with the current mRNA.
Yeah, so there will be like different proteins will be required. All the working in the human body is only proteins.
So the enablement of this splicing process is also a resultant of like a protein function. Okay, cool.
So if there are mutations in that protein itself, so now the alternative splicing can also be modulated. So you see, there are various aspects of looking at alternative splicing.
So can you speak like more about the focus of your work on splicing? At what level are you trying to design an intervention, or what are you studying exactly?
So this is actually still a work in progress. It was one of my project research grant where I actually wanted to look at the structure of this. We call it as a spliceosome machinery. Everything we convert it into machinery because those are megadaltons, bigger proteins, larger number of proteins, they come and go. So the thing is that the trick is it's a very conformationally dynamic molecule, or I'll say the whole of the machinery.
Right.
So many proteins, they come and go, come and go, come and go. So my task is to look at or connect the dots that we know that there is a cycle which goes on. What else we know till now which is experimental. And my part is that if the computation, because the experimental is going to give you which you can actually see it. It should be longer lived. There might be conformations which might be transient in nature, and they can be only easily decoded when you have a computational, because now you can go to in your femtosecond scale or nanosecond scale, and now I can create or link those missing experimental information with the computation studies which I have done in the spliceosome machinery structure. So this is work in progress. It requires lots and lots of things because there are so many bottlenecks that happens.
So that's a sophisticated machinery to just like be able to splice.
And every time if you find some partner, for example, let's say if there are 10 conformations possible in the cycle of a spliceosome machinery, cell might be doing it faster over millisecond or something like that. For us to actually compute it, it will require lots and lots of computational power because the number of atoms, the partners, they are going to be larger in size and they are quite dynamic in size. If they are coming in stage A, they won't be present in stage D. So that coming and going should be accounted. So that accounting part, when you want to model a system which is as dynamic as nature, that actually require quite a bit of let's say runs and trial runs and working with that. So it's a very, very challenging.
So our current attempt is still sort of at just enabling, like figuring out that robust machinery of carrying out splicing. But once we have that, what will splicing enable us to do?
Okay, so the thing is that in humans, various proteins, especially the neurodegenerative ones, so they
Yeah.
are being associated with the protein isoforms. So these protein isoforms are something, the same mRNA is going to give you some protein, right? Now, if you have a different splice junction of it, so you will have a different
Different
isoforms
protein.
of a protein. So that is something that if some of the protein is creating, let's say, aggregation or
Okay.
prion aggregation kind of a problem, so that can be solved when one can tackle that what the alternative
Okay.
splicing are.
But that will also mean that we need to be able to figure what and where do we splice in the RNA. How are we
Yeah,
tackling that?
so
Have we figured that
that
problem?
how we are tackling with, so that is something still everybody is trying to do that.
Okay.
And my knowledge may be limited also here also, because I am not primarily a very experimental person in alternative splicing. I am working on whatsoever experimental data that is going to be there, and
I
then
see.
linking with respect to the computational, so that I can complement what the people know in alternative splicing. So the data, the information that I know is based on whatsoever work has been done till now, and I'm pretty sure because alternative splicing has kind of attracted so many researchers worldwide, very
Yeah.
good and large research groups, they are working on it. So they might be on a much more frontier of it
Okay, got
compared
it.
to me.
Yeah.
So yeah.
Okay, let's zoom out a little and just why is the protein so important? What is it doing in the body?
Everything.
Why are we why are we running after it?
Because everything is doing, it's making you good, it's making bad. I mean, it's everything is doing. If you want to digest something, it's a protein.
Okay.
If you're not able to digest something, it's a malfunctioning of a protein. And there are so, I mean, everything, every worker in a body, the small machines that work in a body is proteins. Your enzymes are proteins, your converting from one form to another is a protein. Breathing, oxygen which is delivered to each and every cell of your body, that's a protein.
And so the protein is basically composed of, like, the building unit is amino acid. And
Yes.
one of the biggest, I think, like, challenges you're grappling with is the protein folding, which is like a sophisticated way of just like folding amino acids.
Yeah, that why it's what is the slight kind of luck we have is we have tried to look out that looking, or I'll say it's getting an information about for the observation. For example, if we know that there are 300,000 structures available of the protein experimentally, out of the 300,000 there are like 10,000 or maybe more than that, but not up to 300,000 folds are there. So that means inherent structure is
Okay.
kind of similar for everybody, for some of the families. So we call it as families. We can actually
Okay.
categorize into families. That
Okay.
makes things a bit easier. Okay, so it's something like if a protein is going to do an activity which is a protease activities.
Okay,
One
what is
protein is
that
going
activity?
to do. Protease is like cutting your proteins and creating a chunk or those kinds of protein-protein interactions, kind of. Say, I won't say protein, I would say it's a bond, it cuts the bond. Proteases.
Okay.
So they cuts a bond.
So that's a kind of family?
Peptide bond.
That's a kind of family?
That's a kind of family. Kinases, so they are one family, which are like phosphate bonds, phosphatases who add or delete the phosphate bond. So there are different kinds of families which are available.
And so within the families there are certain
So there
similarities of how the
there are
folding
similarities
works.
that are being seen.
Okay.
That we see that
Okay.
there are similarities that happen. That means the inherent part, the way they work, they are similar inside within the family itself.
Right.
So that actually sums down them, categorize them lesser to work with. So we see that if a thing has a function which is a protease or a kinase function, our first thing will be to go and check whether we have similarity of it with the other proteins which do the similar work or not.
Okay.
And sometimes it helps also that we can create homology modeling and all, and that is why you see the AlphaFold, which is
Yeah, that's
quite in
the structured
news.
prediction.
That's the structured prediction.
Why is structure prediction a good problem to work on? What will we be able to achieve if we get really good at predicting the structure of the protein?
Okay, so the idea is with respect to this that if we know the lock, we can know the key. If we are concerned about that there is a protein which is creating havoc in the body and it's not let's say a very healthy
useful protein.
Healthy protein.
Yeah.
Or is resulting in a disease state.
Okay.
So what you can do is that you can actually create drugs for it. Okay. So mostly the enzymes are the one which are responsible for creating that part. So they have a particular structure to do it. Now the structure is something, for example, let's say if you have a scissor and you want your scissor not to cut anytime, what you will do? You will either bound it or you keep it in a state so that it remains inactive.
Okay.
So that's what we want with the proteins to do. We can put some drugs there, or we call drugs, or let's say the ligands we call, or inhibitors or something, so that we can inhibit his function.
Got it.
And if it's inhibited, so the disease state is less.
Got it.
So that is to look at the structure of a lock.
So, okay, so the prediction problem is, the structure prediction problem is meaningful because once you know the structure of that particular troublemaking protein or disease causing protein, you can then come up with interventions to sort of intervene and make that function differently.
Yes.
Okay.
So one is with respect to this, which I am saying disease. The other is with respect to enhancing the protein. You can enhance proteins too.
Okay.
I mean, for example, in lipases.
Right.
So lipases are something which are going to cut the lipids.
Okay.
Fats.
Yeah.
Lipases are also ingredients of our washing powders.
Okay.
What it does is that if you have a grease stains, oily fat stains, so it actually cuts down those with the fiber and then it releases out. So lipases are the one which are involved in so many industries also, based on wherever these fats and lipids are being used, and they need to be processed to that time. So this bacillus subtilis lipase is something which is mesophilic. Mesophilic is something that is like working at room temperature. And you have seen that whenever you'll see there is this what do you call detergent. If you look at, they always say for like very tough stains, put it in a detergent for like no, put it in a water, maybe hot water or at 40 degrees something.
Yeah.
Right. So that means what? That means the optimum temperature for that enzyme to work with that structure will be 40 degree.
Right.
Okay. Now let's say if I want to work in a place, or let's say I want to make, there are certain things, for example, detergent making industry, you want it to survive at 50 degree and 60 degree. Within organism, nature won't care whether that protein works at 60 or 70 degree.
Okay.
It's only going to care what that organism require.
Got it.
And organisms stay at 37 degree. So the enzyme will work at 37 degree. But being human, we want to make it better for our use. So what we can do? We can induce mutation. Once we know a structure, we can induce mutations also.
Right.
And once we mutate, we can see that how the efficiency of that protein or that enzyme can actually work at high temperature too. So now you can modulate. It's all about playing with the modulation.
So would it be right to say that if I am able to put a finger on what are the proteins that help in digesting fat quickly, and then will we be able to just like create drugs that help people cut down weight very quickly?
Yeah, I mean theoretically everybody will be like, oh yes, that can be done, but in actual it does not happen like that.
If you're able to like make like solid progress on this, you can just like re-engineer the whole
re-engineering
body.
is what we have been doing like from day one. Okay, so for example, you might have seen, I don't know, glowing bunnies, glowing
I've come across
what do you call, jellyfishes and all those kinds of things, right?
Yeah.
Jellyfish was glowing from since it was born, like say, like GFP protein that was already discovered in jellyfish.
Okay.
But we actually modified to create so many colors
Right. Yeah.
because we like to modify things, we like to modulate things.
Yeah.
We humans like to engineer for that part.
Yeah.
Yeah, so digestive enzymes are something that everybody wants to work up with, how the metabolic syndromes and everything, it can be kind of taken care with digestive enzymes. But there
Okay.
are so many integration of other aspects when it comes to actually working. For example, like I told that protein structure, if you know, you can create drugs,
Yeah.
right? I may show that this drug has a very nice affinity.
Yeah.
But on the other hand,
let's define affinity.
Affinity means it can bind there, it is not going to leave it.
So it
can
bind,
inhibit it.
It
at
can
that
bind to the protein in a solid way.
In a solid.
Okay.
It is not going to leave it.
And
it is
then
just going to keep bind there. Okay. But the thing is that when you go from that computers to as soon as increase in, let's say, to
Human-like
rodents
conditions.
models, then you go to humans. So
Okay.
all those things.
So,
Affinity
so
changes.
there's a very interesting aspect of like all the complexity that's involved, one in drug discovery, but then like drug delivery is the other main
Yes.
like challenge that comes.
Drug delivery is one challenge for
Right.
that.
Yesterday I was speaking with someone and I was mind blown to find out that like the design of the tablet, like the shape of the tablet is actually a design decision and not like random. So if it's like a tab or if it's a capsule or the shape,
Then how it get easier to go through it and then absorb and other things. Yes, true.
It's fascinating.
Yeah.
So since we were starting to touch on disease and drugs, a lot of the diseases I believe are like a function of mutation, right? Would that be right to say?
Mostly mutations are there that leads to those diseases, but sometimes it's not even the mutation, sometimes it's accumulation also that can happen. Proteins
Accumulation
can
of
get
what?
accumulated one on over the other.
Can you unpack that further?
So it's something like, of course, mutation part is that, and sometimes it's the chaperone which actually kind of fold the protein. Protein has got misfolded.
So
they keep on kind of. So you see the principles that I told before, the physical principles, the attraction principles, right? So there are charged molecules, they are going to either attract or repel each other.
So these things,
hydrophobic
the chaperones are
interactions.
basically helping the amino acids fold into the protein.
Yeah, they are one family of, they are family of also of a proteins who help the other proteins.
Got it.
So there's.
So the disease is always a function of the protein sort of not folding in the right way, and there could be several reasons for that. So mutation is one
part,
and the other one is that the chaperone's not working properly and like proper, maybe like
there
environment
can be various
conditions.
aspects associated with one disease itself. It's not like there is one-to-one correlation always.
Got it.
There are multiple things also. There can be one-to-many things
Got
we
it.
see what is happening now.
Yeah.
But what is actually happening, it's a kind of what do you call this, multitude of different things that is going wrong.
It's also a lot of like the function of the environment in which this is happening.
Yes. I mean, I am very much interested in working this human disease progression, but still I'm just starting off the journey with the human disease progression. So I'm also a bit very, you'll say, the beginner in this.
Can we touch on the drug discovery process, and then from there we'll also go into, and because like one of your areas of research is computational drug discovery. So then we'll go touch into like how's the computing helping accelerate the drug discovery process?
Oh yes, that's fine. As let's say I have found a small molecule, a compound.
Yeah.
Or let's say even if it's a small what do you call, peptides,
right?
So oligopeptides, so anything which you take in and that is going to be inhibit or enhance, those are called drugs.
So inhibit or enhance the function of a protein.
Yes.
Okay, so the drug is always interacting with the protein.
Yes.
Right. So the first problem is also,
some drugs can interact with your DNA too.
Okay.
They are intercalated drugs and they are actually poisons. They can go and bind in your DNA itself. so they can create. Even some of the proteins can work with the RNA as well.
Some of the proteins,
Not
but
proteins,
we're talking
the
about
drugs.
drugs.
Sorry, the drugs or some of the entities of, I won't say these are called drugs. This was, I think, a couple of years, not couple of years, few years back. So there was what we call as siRNA technique.
Yeah.
So the same thing which you said with the mRNA part, right?
Mm-hmm.
So these siRNA are small molecules which can actually go and you make the sequence similar to your mRNA, which is there, or not similar, I say complementary to it. So now it has found another partner, it is going to join it. As I told that the mRNA is a single stranded, so it is now converted into double stranded. So what will happen is that cell doesn't like a double stranded RNA. Cell utilizes your mechanism. Whenever it sees a double stranded RNA, it will chop it off. So theoretical wise, everything looks perfect.
So that then that stops the protein
mRNA,
formation, protein synthesis process itself.
At the mRNA step, but the success is still, I think, still to be seen.
So, but
for smaller molecule, I think it was done. Couple of studies were there. I'm not recalling that, but it came out as a very hot topic at that time. That siRNA is actually is a very nice drug. This delivery techniques to be working with, which can take care of all the mRNA which is resulting in a protein or disease protein, or let's say mutated protein, or whatsoever you want.
Got it. So we were speaking about
drug discovery.
Drug discovery. Yeah.
So these all aspects are actually part of drug discovery.
So your first thing is going to be about finding the target protein which is causing the disease in the first place, right? And there you
or the reason that why it is causing the disease. If you want to convert everything into a drug discovery, so the thing is that the main thing is to find out what is causing that state.
Right, and so that would mean basically attributing that state to a protein or a group of proteins.
I'll say group of biomolecules.
Okay.
Most of the times the proteins are because they influence those biomolecules.
Got it.
So that's why you believe that
and again,
they are going
so
to
the
be
intervention will be at the protein level, but the interaction will be with the biomolecules. So how do we find the right, like in the process of drug discovery, how do we find the right target protein?
So there are ways. So experimentally it can be done. For example, let's say you set up a plate and you know these are my different kinds of structures of compounds that I have. So you have, let's say, a shelf of 10,000 million of compounds.
Okay.
You take a plate of that protein or the whatsoever state you find that this is going to create that disease. Just put one by one and try to look whether it makes it inactive or not. Look at the activity of it. That's an experimental part. Experimentally you can do that. But the thing is that this creating of the actual hardcore library of compounds and then doing it within your experiment is going to be, first of all, costly affair.
Yeah. So if I'm not wrong, you're now talking about the molecule that's going to be interacting with the protein.
Yes. Or even the peptides.
But I'd like to take a step back and go to how are we finding which protein do we have to target?
Okay, so for that thing there are various stages that goes through. So within central dogma you see there is a DNA to RNA to protein.
Yeah.
So the things that can be studied is, let's say, genomics, DNA to DNA part. DNA, you try not the actually if you look at the DNA, but how the sequence look like. Okay, are there some changes in that sequence which is compared to a healthy individual?
Okay.
So one part is that. If yes, it's a genetic disease.
But how do you know where you have to look?
So that's what I'm saying. You map everything. We already have a DNA human genome already done.
Got it.
Okay. And for example, the idea of getting that part that let's say it's something, what do you call, if somebody is hypothetically is not able to, let's say, which example should I give?
Let's say if someone has a speech problem, they stutter.
That's a lot of things. That's not only disease. It's not a disease.
Let's take a simpler example then.
It's not a disease. It's a very complex phenomena. It's not only just related to that person, this thing. It's the physiology in play.
So that's not just a genetic, a gene function. Okay.
Let's say a gene function is somebody is not able to digest fats.
Okay.
Fat digestion itself is a very after the least amount of thing. Let's say if given chances
Right.
for our body to eat, fats
Yeah.
are the one which will be taken up last. That's why they get accumulated.
Yeah.
Okay, they are the last one, but body can do it. It's the last resort. So keto diet and everything is like
Yeah.
the last resort to do it. Okay, so that's why it gets accumulated. Let's say somebody has a problem with that. So that person will have lots and lots of cholesterol, lots and lots of lipids
Okay.
in his bloodstream and everything.
Yeah.
So now you have something to look at. Okay, there is something that is going on. So that profile
Yeah.
is going to give an idea,
Mm-hmm.
kind of. Now you want to pinpoint it. So they say, okay, what was your family history?
Mm-hmm.
So they are going to look at whether their family history have it or not. Somewhere
Okay.
they have it or not. If they say yes, they
Right.
also had the similar problem.
Right.
So they are going to look, okay, then why don't we look at the genetic profile of you? So they are going to get their genome sequenced. And once they are going to get the genome sequenced, they can actually look at which are the actual region of the genes which can create creating a problem.
What are sort of like the unusualities in their genome?
So our genome is made up of only 4 things.
Yeah, but
ATG,
the sequence,
DNA.
like
The sequence will be different.
unusuality in the sequence is
Yes.
what they'll map.
Yes, so what are the
Okay.
actual things that were changed?
Right.
For example, instead of adenine, that person might have cytosine.
Right.
So those kinds of functionality it will match
Right.
with whether it's a genetics or not.
Right.
Once you've done with the genomics, that's the genetic part. So
Mhm.
that's another. So there will be another line of therapy for those cases.
Mm-hmm.
Now, if they say no, there is no difference in the genomic part or something.
Yeah.
So then what they are going to do look is that they are going to look at transcriptomics and proteomics.
Mm-hmm.
So they are going to take the serum. It will have all the proteins. Let's say they take the lipids from there. There are many ways to do it. I even don't know all of them. But let's say if they take it from the serum, those proteins, and then try to digest it or try to look at what the sequence of that protein looks like. It's a small, is of the similar length or not? Does it form aggregates or not? So those kind of biochemical techniques can be applied at the proteins level. Now they have identified whether this is a protein or not.
Hmm.
Okay, and then when they have identified, they can also link with the mRNA as well. Because when you know the DNA, you can convert exactly into mRNA. Right?
Yeah.
Now from mRNA you can convert into protein. But when you see the mRNA, when you try to convert into protein, it does not get into protein. That means sometime it's splicing somewhere around.
Right.
So now you have linked it with the splicing itself.
Right.
So there are various steps.
So that's how you're able to pinpoint where the problem is happening.
Where the problem is. The other part is that because your protein is enzyme,
Right.
you can also look at the metabolics of
Okay.
it. Because those are the products.
Right.
So that can also tell you that protein might be folding in the way you want it. It is not aggregating, it's not doing something, but it is not making the products the way it is supposed to be made. So you have metabolomic studies. So there are various aspects, the integration of this study which can actually help you to pinpoint that this is what is happening. It has, you'll be amazing to see how the biological approaches has come.
Right.
From, let's say, from the century or beyond it.
Okay.
It's like it's a very, very long way. We have come quite a long way in that.
Yeah. So let's go back to our drug discovery problem.
Yeah. So that's
we have the target finding.
Now we have the target
and
finding.
then we have a molecule that we're designing to come and interact with this target.
Okay. So I'm going to come here back to what I said about the experimental. You can set up all the plates. You can look at how they are interacting, and you can look at whether your enzyme assays are now activated, deactivated. You can do all sorts of things to see that this is happening. This is now working.
So this is
on
an
it.
extremely hit and trial sort of approach.
Yes, it's like applying everything to this.
Okay.
If you don't know anything about it.
Okay.
Okay. The other part is that if you know a slight amount of it,
Yeah.
and you can find the, for example, the homology, or let's say the kind of similarity to the other proteins which are already known.
Right.
Then you can create a structures. You can get a structure, and once you have a structure, then you can use it for a computational drug designing. That saves the money. It's like the first line of things. So it's like you can take a 1 million of compounds. You can keep on scanning one by one. You apply, and then you look, okay, based on the interaction of this protein and the ligand, what is the energy or the score I'm getting? Is a good or a bad one? Then remove that. Then the second one. Then the third one.
Okay.
And relative order of it is going to tell that which are the good binders, which are not so good binders. And the good binders, you will feel that if it's a binding score is good, that they are going to be having a high affinity.
Right.
For a
Okay. So
protein.
binding score is basically mapped to affinity.
So now what you have done is that you have found a seed to work with.
Okay, like a seed molecule, seed compound.
Yes.
Okay.
So then you can take those hits
And
or the
then
lead
you
compounds.
can then further like play with that compound, with that base.
You can make changes.
The seed shape.
Theoretically also you can make changes, for example, add some function groups here and there and try to see whether you can make it better or not. And the second, after doing that, instead of getting for that 1 million shelves, now you are going to look at one shelf only.
Right.
Because experimentally you have to validate it. It's always the validation that.
So the main part that we've been able to accelerate in the drug discovery is finding the molecule that binds with the target site. That process
that's what
we've been able to
once,
accelerate. Right. Okay.
Binding site. Yeah, let's see. For example, binding is also in different modes. There are other things associated with conformationals with respect to binding as well. So the first instance, we are saying that if it is accepting that molecule, that means it is allowing to get into that site. Sometimes the drug molecule bind in some other site as well. So there are multiple mapping that needs to be done.
So that
for drug
making
design.
sure that it binds to the desired target site and not to like other sites. One of the things I was thinking is, so in the drug creation process, the compound is always going and binding to a protein, right? So given this model, the nature of the cure is it's not really stopping from creating those misfolded proteins. So then the nature of the cure, how resilient is it? Because the way we are sort of treating this is we're always going to have to keep putting in that compound to make the protein function in a similar, in like a different way. Why have we not, have we looked at treating the misfolding itself?
Yeah, so there have been instances where people have done some experiments to work with misfolded proteins.
Okay.
So misfolded proteins won't be like, let's say, a drug or something. So what can happen is that all the proteins can be marked or flagged to actually chop down. So if that can be worked around, so mostly cases like you can work at RNA level at that point, or it's harder to work with misfolded proteins.
Okay.
And harder when those proteins are in your brain. So let's see. So what happens is that even I'm not very much into those misfolding yet.
Okay.
Folding and misfolding proteins, but yeah, so there are therapies which are with respect to how the folding of a protein can be taken care of. It can be actually associated, or let's say marked with some of the things so that it can directly go to the protein degrading machinery, because there are there too. So it can be simply chopped up as soon as it's misfolding. Cell does it too. For example, if somehow the protein has
a mechanism.
It already has this mechanism that they can be misfolding occur. So it's going to do it. The only thing is that when either that machinery is a bit slower, or when the production of that misfolded protein is larger. So then it's going to hijack.
Then it starts accumulating
that it's
the misfolded protein.
Yes.
Okay.
Otherwise, cell has its own mechanism to take care of. It's a smaller in is going to be gone to that degrading machinery. Everything is recycled in cell. That's the good part.
Got it. Let's go to AlphaFold for a moment. And so AlphaFold is basically predicting the how the protein will fold, the structure, basically, right?
That's what we see.
So what is the anatomy of the training data for something like AlphaFold, and how are we combating? So also, given the fact that so far we've only been able to fold a few molecules, right? So how do we manage the problem of like very few data?
Well, AlphaFold is a good thing to work with. I don't want to comment more for it. There are so many partners for that, third included. Anyhow, so the thing is that AlphaFold has its own advantages in many aspects. Okay, because even for a smaller data, it is able to accurately predict many things. Some of the data are very specialized. If there is no other information available for those, then the use is still, we need to see. So what happens is that there is this CASP, we call this competition, which is a critical assessment or for structural protein, something like that. So they are the actual, it's a kind of competition, which will happen is that people will create a structure for it, crystal structure, which is the experimental structure.
Right.
What it will do is that it will give a sequence.
Okay.
And everybody who is working in the field of, creating protein structures, ab initio, or they have their own softwares to create.
Right.
So they all enter that competition and they try to create the structure as close as possible. So it was amazed that when AlphaFold first joined this CAS project, so various chunks of the proteins it was able to do very good, but some of them it actually failed.
So the sequence here means the amino acid sequence.
Amino acid name,
and then you also have how the folded structure is going to look like. So you get these two data points.
No, the thing is that you don't get folded structure. All you get this 20-letter code of amino acids.
Just a sequence.
Just a sequence. It's a text sequence.
Okay. And
then they say convert it into a structure. They already know how the structure will look like. And when the results are
the closest gets to win.
To win.
Okay. So I believe like the model takes from like how like within the 20, like how a specific sequence sort of interacts with the neighboring amino acids and like things of those. Okay.
True.
That's fascinating. Awesome. Let's switch gears a little bit and maybe talk about your journey. So how did you get into computational biophysics? Was there like a moment of magic?
I was intrigued by the enzymatic structures.
Yeah.
I was intrigued by the I'm a chemist.
Yeah.
So
you mean you started as a chemist?
Yeah, I'm master's chemistry.
Okay.
So yeah, I loved chemistry at that point.
Okay.
And the thing was that I even loved theoretical chemistry, quantum and all. So in a sense, but then I was intrigued by the enzyme structures. We had one course and there we looked at the catalytic triad. So 3 amino acids, they always come in kind of a similar orientation to create something new. Okay, and I was amazed at how nature actually does it, because for us we can see. For us it's like, oh, always this is going to be H, this is going to be A, this is going to be D, this is going to be B, that's why it is going to do that.
Yeah.
They are hydrophobic, that's why they are going to do it. They are hydrophilic, that is going to do it. We provide physical principles for that. For in actual 3D molecules, what's for them?
Right.
It's just their interaction.
What intelligence do they have that they're repeating that behavior?
They're repeating that behavior. They don't have a thinking that I want to eat this, I want to get this, why can't I get something else, why I have to do this.
Right.
So that was something that amazed me
That's
at that
fascinating.
point. Yeah, so I was like, okay, I want to work with proteins because it seems to be more chemistry in nature. I'm going to do that. Okay, fine. So then I applied for PhD in proteins.
Right.
So I wanted to be a protein crystallographer at that thing. More towards experimental part, though I know nothing about biology at that, nothing. I just knew about chemistry.
Okay.
Okay, and then of course after that, by fate or something, let's say then I had to change my PhD because my guide left for some other institute, and then I changed to IIIT Hyderabad, where I was with computers.
Oh, you see, it was it was by chance that
It was by chance with computers, right? So then, yeah, so then it was like, since I was intrigued by structure itself, and I said, okay, I can do biology in computers as well. So then I learned many things, and of course, so then I went into computational biophysics because then I could see it.
Right.
And experimentally, the problem was that you can't see it.
Yeah, so you just
So,
got more and more tools.
Yes,
to...
the resolution, the atomic resolution, which I can do, and theoretically, what can't I do? That's the issue I had.
Right.
I mean, I can say them, okay, you want to see splicing, snip this out, I mean, cut this out, and then
you
can see it. I can see it.
Actually getting
but the thing is that if I want experimentalists to do it, what experimentalist will see is that I do it, but the organism die. I don't know what is happening. So you see the end product of it. You don't see what is happening inside.
Right.
So that made me quite happier that, okay, I can see what is happening inside. I can relate what is happening inside.
Right.
Yeah, so then that came. So this was mostly into structural biophysics. And when you are working with structural biophysics, often to answer many questions, you have to go into bioinformatics, employ many techniques on the go. It's like a field which is like one of those fields which cannot sleep.
Right.
I mean, you just keep on brushing up your skills. You need to brush up your skills for each and every aspect that comes to you. Okay, for example, if I see that this is going to be happening, can I apply the learning method for it or machine learning to it? Can I get AI component? So this is where I'm trying to go into, I mean, currently as well. So there are various aspects which are there in computational biology.
Right.
I mean, it's very vast. You can work on each and every aspect of it.
Yeah, but like I like the idea of like how you're sort of like going deeper into the journey started from that sort of like just curiosity about like how the hell do these like machines work.
Sometimes it's something like I feel myself as like people say now, God wants you or the fate wants you where you want to be, not where you need to be.
Yeah.
So it's something like,
yeah,
I'm here. Somehow I started from something else. I wanted to do something else, but I'm here and I have no regrets. I'm happy for that. It turns out good.
Yeah. What do you feel are like the, like if you had to put like, what are the moonshot possibilities you feel like, or you know, that are near, that inspire you to be here and like keep doing what you're doing?
Yeah, so the same thing which I told, that I'm a beginner in human disease progression. I really want to see where I go. So my aim is to actually create various things with respect to that nailing that actual problem of how the disease actually progress. So one part is I can now see that from where it can be originated. Okay, and let's see from where it can originated, the kind of thing how it can be elevated. For example, with this
with the
drugs and all, how it can be taken care of that.
Yeah.
And then afterwards is like, once it progress, can we look at the progression of it as well? Okay, so it's like from the starting to the end.
Yeah.
So where I can help.
So when you say look at the progression, what exactly do you mean there?
Many diseases are like age related too. Okay, so somebody might be healthy now, may feel it, or so there are various aspects with the disease. It's not just the not just the folding of it, machinery of it or something like. I mean, there must be something that is relating to, let's say, the progression of a disease, and it's also individual dependent too. So there are various aspects that goes into personalized thing.
Right.
And then, I mean, I don't know how I'm going to do that, but that's what I want to do.
Okay.
So it's simply like Moonshot.
Right. So that's like, so.
It's just like I want to do it.
As like a. So as a function of aging, let's say, so we can think of disease as just like an adverse condition. So aging in itself could somehow be like framed as like a disease. So you
okay.
Would it be like a cool idea to think of like that we're trying to solve for aging, because aging is a disease, this basically, right? Like, because like bodily functions are like slowly degrading.
Yes. So the thing is that why they are degrading. So there might be various, you see the multiple things that is leading to one, right? So one to many kind of correspondence.
Yeah.
Yeah.
So, but I'm sure that you're gonna have some models to like, why is this happening? Just, like, I'm sure like you think these are things you probably like spend a lot of time thinking. What are some of those rough uncut models, like why does aging happen?
I have no idea yet, because aging I still have to go.
But like, just even for progression of disease, do you have any ideas?
Well, somewhere it's something that's what I'm saying. There are various aspects for that. For example, aging can be related to whenever you have a new, at the cellular level, it will be more like more cell death. New cells are not able to born.
Okay.
So that's another aspect for that. And people have been doing various things to look at. For example, the one who got like Nobel Prize for telomerase activities. So she also looked at that when the chromosome division happens, the size of the telomerase, the end of the chromosome, they get on decreasing.
Okay.
But this is also related to stress too. So more stressed people seems to age more.
Right.
I mean, that's also one line of thought.
Okay.
I mean, there are still many things which are at the theory level.
Right.
But experimentally, or let's say with the concrete scientific proof, that needs to be looked into.
Right. And like, if we just think of it as a thought experiment, like, it should be possible to design a mechanism that helps us sort of like break that relation between aging and the length of that particular.
That they have done. They have done that. Yeah. They have done it. They have tried to look at it, but then they have found that there are other aspects as well.
Right. Why do you think there are so few people in the field? Is like a lack of awareness, or is there a barrier to entry?
There can be various aspects of various fields. For example, the kind of commitment it might take. People now are kind of that, they are now open to the things.
Right.
For example, the first time when there's been a story going on, the first time the human genome was actually sequencing project was kind of introduced, and they said that they are going to need computer scientists, and people were like, what the computer has to do in biology? Now the people are open.
How long ago was this?
I think that project started about 1960.
Okay, that was a
somewhere
while ago.
1960 or more than that.
Okay.
Before that.
Okay.
I'm forgetting the date.
Okay.
It has taken lots of time to actually come to where we are today.
Okay.
In sequencing as well.
Right.
But that project was started way back.
Right.
So, in the sense, I won't say there are many people who are not here, because biology have lots of people.
Yeah.
Right? And every now and then, the only thing is that they are not aware that they are doing computational biology as well. For example, they design primers.
Right.
They are using computational biology.
Okay.
They are creating their statistical, let's say, graphs to show that this is significant, that data is significant, this is the n number of observation. So they are applying statistics to it. They are using computers for that. So that aspect is also there. The only thing is that the kind of input it takes to actually create this field is that you should be having information about biology.
Right.
It helps if you know how the experiments does.
Right.
What are the ins and outs of those experiments?
Yeah.
And then, of course, the physics, because you should be knowing what are the formulations that are being used, underlying. Computational tools, you should be knowing programming and computation. And since everything is going to be averaged out, statistically looked at, so mathematics. So it's like there is a combination of all these projects, which actually people get scared. Oh, I have to study this. So maybe that's why.
Right. Yeah.
I mean,
it could sound like it's
but but currently it's changing. Looking at how the AI is also helping and shaping it up.
Right.
So yeah.
Right.
So people are now.
Let's switch gears a little again and like speak about your current role. So you're an assistant professor at Plaksha, and you're sort of heading the biosciences undergraduate program here.
Program coordinator.
Right.
Yes.
Yeah. So can we reflect on your experience here as an educator?
Yes. So here at Plaksha, the BSc, the curriculum, it's quite new. Okay, quite unique compared to in India. Because in India, you will see these kind of curriculum actually get introduced at the master's level, PG level, not at the UG level.
Yeah.
So the tougher part was to actually bring out biology.
Yeah.
And give a flavor of biology as from the engineering point of view, right? And when you are a biologist, you get more into this basically and fundamental and this thing. Engineering approaches are different than scientific approaches. They are a bit different from that. So to actually make a combination of both and deliver in such a way, and actually to an audience, the UG audience, who are from this IT kind of or CS loving background.
Yeah.
That seemed a bit trickier at first.
Okay.
But now it's quite enjoying because currently I'm seeing biology from altogether different point of view.
Yeah.
I saw it from chemistry.
Yeah.
I saw it from the computational. Now when I'm seeing from the engineering.
Right.
So it's amazing.
More and more modalities, and they're creating richer models.
Yes.
Right. Yeah.
So it has helped me as well also to go as an educator.
Yeah, that's fascinating. But what was the philosophy behind bringing this program at an undergrad level?
So this was that many healthcare industries are there. So they are coming up as startups where they need. So there is a demand of also let's say so many people who can actually be a problem solver,
who can work at the interaction of these interdisciplinary fields.
Yes, so they can work as a bioinformatics, or they can also work as a database management, or providing solutions, or even at the genomics level, various R&D labs. So there is the market which is going to be there.
Right.
And also we need engineering solutions with respect to biology as well. So those kinds of thinking should be there, tinkering in biology, because the healthcare industry actually need. We need various perspectives to answer, let's say, even one question.
Yeah.
Yeah.
And so, I feel it's also like such a meaningful work to sort of help develop like young people the curiosity into how all of these like insanely complex but beautiful machines work. So challenging but also meaningful. What does a day look like in your everyday life?
It's the normal day will be like I'm a mother of 2 who are school going, so that's it.
You get them prepared in the morning.
Prepared in the morning, their lunch, and then send them off. And then afterwards, sometimes drop them towards school. And then afterwards, then I come to Plaksha, and then they routinely all classes, academic session, meetings on so many levels. And then there are so many projects which are going on, research projects, those meetings. And then I have even collaborative projects. So yes, so it gets super busy, and then I just drive back home. And then it's like totally tired at the night.
Nice. What would be like your advice to young people?
I mean, my advice is that I don't know, people are something, why we have to do this, why we need to know this, why we have to do this. I mean, for me, it's like life is full of surprises.
Yeah.
Sometimes you even don't know that the things you don't like, you
Yeah.
actually end up doing it. So my thing is that just be happy and keep open to many things that comes your way.
Right. Yeah.
Yeah, sometimes I have so many students coming and say, why we need to do this,
We
why
tend
we
to
need
complain.
to that, why we need this course, why we need that course, or all those kinds of questions.
Yeah.
It's like,
so, like for you, you were able. At what point were you able to let go of that complaining nature? Like that's very common.
I
like,
never had that complaining nature.
Okay.
That's what I'm saying. My thing is that if I've got something in my hand,
Okay.
I just need to do it good, give my best so that I can sleep better at night. That's
Okay.
the only thing.
Can we reflect on like moments of challenge and difficulty in your journey?
Okay. So yes, so the first one was with respect to learning biology.
Mm-hmm.
I can still remember I was having I think it was second or third day of my joining in tech. They had this conference of somebody so renowned, but I remember that name because I wasn't aware of biology field at all. They were throwing things like, we did STS page, we did this gel electrophoresis, we did that Western blotting. And I mean, today I know about it. At that time, I mean, it took me 1.5 hours or something to know about that,
Yeah.
what it means. Not even years, but like months of that to know about what is that. I was just looking at his face, but he was talking about so many things, and it was just going above my head.
Yeah.
I mean, learning everything. The pre-PhD program that we had in MTech was GNU. At that time, it used to give GNU degree. I don't know now. So that was a very intensive program,
Yeah.
especially for somebody who is non-biology.
Okay.
But then I had to study so many books at that point of time
Yeah.
to understand
Okay.
each and every aspect of that.
Okay.
So then we had to be at par with everybody, right? There are people who know biology like to the end. They have done biochemistry for their 5 years or molecular biology for 5 years, and I'm coming something who have just taken one course in biology, in some biochemistry oriented, and that's why she was excited about it. But then but I had a very good batchmates at that point, so helped. And then I like to study more and more biology and learned, and then did it. So that was first thing when I learned biology. And then all of a sudden, when I have to chunk everything out, learn computers. That seemed harder to me at that point. But it was fun learning programming.
Yeah.
Though it took lots of time.
Right.
My aim was that, I've already done 1 year of my PhD. I've left 1 year less to complete it.
Yeah.
I mean, I was counting that, okay, up to this point, I should have done my PhD.
Yeah.
So that was a bit scary to learn it, but it helped a lot.
Yeah.
And then learning how to create those servers, combining different computers, NFS file,
Right.
NFS servers, right? So I learned those kinds of things from there.
Yeah.
And yes, so that was actually, but now it seems that was very scary moments at that point, but here it's fine.
Yeah.
The other later scary moment was when I actually came from US to here and I had nothing in my hand.
Okay.
So there was working with NIH, but when I came here, I had nothing.
That was
in my
like a
hand.
leap of faith.
Yes, that was leap of faith to try with.
Yeah. Yeah. And I was just reflecting on what you said. Like, I feel like back in the day, it was more challenging to get interested in general, like in all of this. But now, because we have like so many great tools and visualizations and like, I think
So,
storytelling is just like mind blowing now in the field of like biology, computers, and
Information is available
Yeah.
now easily, so it's
Yeah.
really good.
Also, it's not like dry and it's
No.
like really rich, like it
Yes,
gets you
yes,
interested.
yes. We actually had done those chemistry, what do you call, thesis and all, looking at those abstracts within those books.
Yeah.
You had those abstract findings. Now you
Yeah.
need to look at it. How many abstracts till that years you finish, like year wise, not year wise. So it was like 5 years or 10 years binder you had. So whatsoever work has done here in this, go to that keyword and then search for whatsoever work has been done till now and what is required to be done.
Oh my
So
God.
find that kind of missing link. Sitting with so many abstract books was crazy.
Yeah. Yeah.
And then we had like SciFinder, and I was so happy.
Yeah.
Chemistry was almost the first one to get the SciFinder
Okay.
and across
Yeah.
this thing. Our department
That
was
must have
really nice.
required like a god level of patience.
Yeah, PhD usually require that. PhD
Yeah.
usually require that. It's
At what stage do you? Okay, yeah. I mean,
Hmm.
I was gonna ask, like, maybe it's like a call you like for a lot of people, like PhD feels like a big commitment, right?
it is.
Yeah. And so, like, at what stage did you have that moment of, okay, I'm going into this?
Okay, so I think usually that came. I always wanted to go into higher studies.
Okay.
Like to study. The only thing was my parents I had to convince.
Yeah.
Not convinced. They were always with me.
Okay.
But still, is something like we had a placement in our department.
Yeah.
So the company came and they asked that, okay, you can join. And I had already scored that CSIR, but haven't joined PhD yet. Okay, so that was there. Then I called my father and I told him that, look, I have got this offer as well.
Yeah.
But then I also got that CSIR and the money will be here more
Okay.
in company compared to CSIR.
Okay.
CSIR used to give peanuts. Now it's a bit better, but not that better, I'll say,
Yeah.
compared to the inflation that is going on.
Yeah.
But in our time, we had like really
Literally.
less.
Okay.
Literally less.
Okay.
Okay, 8K. 8K was
Yeah.
the thing we used
Yeah.
to get at PhD. I said it's only 8K. But then my father was like, look, the thing is that, ठीक है, jobs आती रहेंगी. All you have to look is that if you want to study more, then I'll suggest don't worry about money. You will get someday.
Yeah. That's,
So money will come to you.
yeah.
Don't worry about it. If you want to study, continue it.
This-
Baki, it's your call. So then I was like,
Yeah.
okay, then I'll study.
How do you define purpose?
Purpose of what?
Purpose of your life. What is the purpose of your life?
I still don't know. I still don't know. Sometimes the purpose is solving, let's say, it also kind of, I think, in my view, depends upon individual too,
Yeah.
right? And also where the purpose is being looked into. What's my purpose of, let's say, with related to my kids? What's my purpose related to that?
Yeah.
For me, as a purpose is, if I'm kind of fulfill everything that is related to others, maybe I'm happy with that.
Okay.
Okay,
Yeah.
so I don't have any this kind of that
Like a
enlightenment area.
big
Whatever.
philosophical
What's the purpose of me in my
ideas,
life?
Purpose.
I mean, even I don't know yet.
Yeah.
I just keep on.
Yeah. But there are
Working, that's
problems
it.
that excite you. There is family that energizes,
Yes.
and so you take you building your life like that.
Yep.
Nice.
That's-
Do you have a final? Do you have a biological joke to tell to the audience?
No, no, I can't remember anything if you ask me what's a biological joke.
I enjoyed our conversation very much.
Yeah,
Thank you for coming and
Me
speaking with
too.
me. And I know you had a very busy schedule, but you still made time.
Yeah, thank you so much for calling me.
Yeah,
Yeah.
I had a ball.
Okay.
Thank you.
Sure. Thank you so much.